Welcome everybody. Herzlich willkommen. Thank you for joining us live today. It's about 11 a.m. in Berlin, 10 a.m. in London, and 3:30 p.m. in New Delhi. I warmly welcome you to this virtual panel discussion, jointly hosted by the German National Academy of Sciences Leopoldina and the Indian National Science Academy. My name is Regina Riphahn, and I will be chairing today's panel. I am Leopoldina's Vice President and Professor for Statistics and Empirical Economics at Friedrich-Alexander University Erlangen-Nuremberg. Today, we will discuss major challenges in COVID-19 vaccination and how these could be addressed from a scientific perspective. Warm greetings to our four panelists joining us from Tübingen in Germany, Vellore in India, and London in the United Kingdom. Thank you so much for accepting our invitation. Warm greetings as well to the many participants connected live across the world. I was told that we have participants from 51 different countries. You may have noticed that there has been a last-minute change. For personal reasons, Ingmar Hör can unfortunately not join us today. But Florian von der Mülbe, who co-founded Curabac together with Ingmar Hör, has agreed to step in. Thank you very much for making this possible on such short notice. We really appreciate it. Before we get started, some brief technical matters. First of all, you can switch the layout of your screen to select your preferred option. To do so, please use the button located in the upper right corner of your screen. Moreover, we want to make this an interactive panel. After input statements from each speaker and a short panel discussion, we will take questions from the audience. If you wish to ask a question, please use the question and answer Q&A feature, which you can find on the control board in the lower part of your screen. My helping hands in the director's room, Ruth and Christian, will collect the questions and feed them into the panel discussion. If you want to assign a question to a specific panelist, please indicate that. Today, we are going to talk about COVID-19 vaccination. We have been in this pandemic for over a year now. It has become clear that effective and secure vaccines against SARS-CoV-2 alongside better treatments are the most promising way out of the pandemic. Several vaccine candidates have already been approved. Many more are under development and in clinical trials. However, we still face huge challenges. Regardless of the specific vaccine type, none will be available on a large scale immediately. Challenges range from production to global distribution and effective immunization programs. Also, we must ensure public trust in vaccines to reach the necessary levels of immunity. Before I ask the panelists for their input, let's start this panel with a poll on our participants' attitudes towards getting vaccinated. It will open up in a few seconds. The poll will be open during the first round of input statements, and we will show the results afterwards. Of course, the poll is anonymous, and it will not be possible to track the votes to individual viewers. We have four outstanding speakers today. Each one is a leading expert in his or her field. I would like to start with Professor Gagandeep Kang. She is chair of the Immunization Technical Advisory Group for the World Health Organization Southeast Asian region and fellow of the Indian National Science Academy. The COVID-19 vaccines, which are currently being rolled out in some countries, have been developed faster than any other vaccine against a new disease in history, with development times reduced from usually 10 years to 10 months. This is truly remarkable. There is now a variety of vaccines and vaccine types available. You have been referred to as India's vaccine grandmother, good mother, sorry, good mother, and feature regularly on Indian TV with respect to the COVID-19 pandemic. So could you please explain what types of vaccine against COVID-19 are available, and are they all equally effective in preventing disease and transferring the virus? Gagandeep.
Thank you, Regina. I don't think we've ever had a disease that has had more vaccine candidates directed at it ever before. I think the record before this was for malaria, and now we have over 300 candidates in development or already developed for SARS-CoV-2. If we look at the vaccine platforms that are available and have been used, you can basically divide them into two types. One is the kinds of vaccines that deliver either the whole virus or parts of the virus. So you're giving proteins and trying to induce an immune response. The second class of vaccines are the vaccines where you deliver the instructions for how to make the proteins. So if we look at the first class, which is where you give the proteins, you can have an inactivated vaccine. An example is polio. You can have a live attenuated vaccine. And the example again is polio or rotavirus vaccines. You can have virus-like particles that take the shape of the virus, but do not have any of the genetic material of the virus. An example of that is papillomavirus vaccines. And then you can have recombinant protein vaccines, and a classical example is the hepatitis B vaccine. If we look at the new classes of vaccines, vaccines that have only have even gone into development in the last few decades, those vaccines include the vectored vaccines, and the first licensed vectored vaccines were for Ebola. There are also mRNA and DNA vaccines, and we'll be hearing more about the mRNA vaccines. The ones that are licensed are the ones from Pfizer and Moderna. And DNA vaccines, there are licensed products that are available for diseases of fish and of animals, but as yet no products that have been licensed for humans. Now, the question you asked me is, are all of these equally effective? And the answer is, it's hard to say. We hear numbers that are 50%, 66%, 90%, 94%. What does this mean? It really means that the vaccines are preventing disease, which is what they are supposed to be doing. No two vaccine trials are exactly comparable, so it becomes a little difficult to say that something that is at 85% is necessarily not as good as something that is at 95%. So WHO had set a bar saying any vaccines that were above 50% efficacy could prevent half of all infections, would be considered for licensure and for emergency use listing. And all of the vaccines that we have so far that have gone successfully through phase 3 trials have data that is well above the 50% bar. We'll need to wait and see how long protection lasts. We'll need to see in much greater detail how effective these vaccines are in different subpopulations, in the elderly, in the young, in people with comorbidities. So I think the message is, yes, we've developed vaccines fast, but we really haven't cut any corners in their development. What we've taken out are the periods of vaccine evaluation that are between the different phases of testing. So we've gone, instead of going sequentially, we've gone in parallel. And so far, we have a lot of data that has been generated that's very encouraging, but there's a lot more that we have to learn.
Mm-hmm. Thank you very much, Gagandeep, for this summary of a very wide field. I will now come to our second person on the panel, which is Florian von der Mülbe. He is Chief Production Officer of Curabac. Together with Ingmar Hör, Dr. van der Mulbe co-founded Curabac in 2000. To my understanding, it was the first company to develop treatments based on messenger RNA. Moreover, you established the first good manufacturing practice production for mRNA worldwide. With the coronavirus pandemic, mRNA vaccines have had their breakthrough. So how do mRNA vaccines work? How was it possible to develop them so quickly? And how are they produced?
Thank you. Also for the questions. As you said, Curabac is now active in this field for more than 20 years. And we are using mRNA as a carrier of genetic information. But in contrast to other mRNA companies, we are not using chemically modified mRNA. That's one of the main differences between the approaches at the moment. So how does mRNA vaccines work? So we code on the mRNA itself the information about the spike protein, for example, and using the natural information process, so to say, that we have in our body in every cell, where the DNA is transcribed to RNA, and this is brought then to the factories for proteins by mRNA. And this messenger mRNA is used for information about a coronavirus. So not the entire virus as described before, but only a part. And it's not the virus itself, it's only the information about this virus or about one protein itself. And this is taken up by the cells and presented to the immune system, so that this immune system can look for infected cells. That's one of the main differences if you compare to this live virus or attenuated virus approaches. So we are giving the body only the information about the virus. And one advantage is, of course, that after that, since it's a natural molecule, the RNA decay is very fast. So after giving the right information, it's metabolized as the normal natural RNA that appears in all our cells. And we can really steer how long this RNA could stay in the cells, and it's degraded and is doing its job. And one other important advantage, and that's why also this development of a COVID vaccine really revealed the potential of the RNA technology, is that it is a real platform process. So that means, as we only change the sequence of the RNA and are always using the same production process, the same steps, the same analytical methods, and so on, we can introduce rapidly new sequences, new codes, so to say, on the RNA, and the production remains the same. And therefore, it's really about revealing the potential of RNA with this pandemic at the moment. That's the reason why RNA vaccines are one of the first on the market, because you can you only have the natural code in the end by 4 building blocks that is changed, but the rest remains the same in the production, and therefore adaptation to mutants, to viruses, or even as we as Curabac are doing it for different areas. So for example, we are also active in oncology with therapeutic vaccination, so to say, or you can encode an enzyme on the RNA. So very different approaches, but based on the same platform technology. That's the power of the RNA technology, to be flexible, adaptable, and very fast in development steps.
Thank you, Florian. Just for information, how many production facilities exist worldwide that can produce these vaccines?
So
like
it's daily increasing, I have to say. We have been since, so we started in 2006 here in Tübingen with the first GMP production site worldwide. And we stayed quite alone for a lot of years, and within this pandemic it pops up meanwhile in the different sites and players, and so it's increasing daily, especially also by our network. So we try to build here an European production network and have strong partners as Wacker, Fareva, and so on. And we are helping to spread the RNA manufacturing knowledge to be prepared for pandemics. So that's our way forward.
Thank you. Let's now move to Richard Hatchett. He is the Chief Executive Officer of the Coalition for Epidemic. Richard Hatchett is a medical doctor and served as advisor to the US government in various functions. In 2017, you joined CEPI, the Coalition for Epidemic Preparedness Innovations. CEPI was founded as a multilateral mechanism with two objectives. First, to accelerate the development of vaccines, and second, to enable the equitable access to vaccines during outbreaks in order to stop future epidemics. As long as the coronavirus can be transmitted, many will continue to be infected and some will die. As the World Health Organization says, no one is safe until everyone is safe. So my question to you, Richard, why is it so important to provide equitable access to COVID-19 vaccines globally? And what is CEPI doing in this respect?
Thank you, Professor Riphahn, and thank you for having me on the panel. I would say the arguments for equitable access really fall into three domains. The first, of course, is the ethical argument. We have life-saving interventions, vaccines, increasingly therapeutics, diagnostics as well. And the idea that these life-saving interventions during a global crisis should only be reserved for high-income countries is really not something I think that anybody would stand up and support. So the ethical arguments, I think, speak for themselves. There's also an argument from enlightened self-interest, and this is principally an economic argument, which is that as long as the virus is circulating anywhere, it is going to cause significant disruptions to international trade and economics. If countries are facing uncontrolled epidemics, they will have to implement lockdowns to regain control of the disease and to protect their healthcare systems, and that will have cascading impacts on the global economy. And so even if a country were able to vaccinate its entire population, it would not be protected from the global economic disruption that we're experiencing. I think the last argument, and I think recent experience is making this argument much more salient for people, is an argument from biology, which is that if the virus is circulating widely in different human populations, it will have opportunities to evolve. And we are observing this in real time, and it is evolving in two dimensions. One, it is becoming more transmissible, and we are seeing convergent evolution in a number of locations. We've seen this in the UK, in South Africa, in Brazil, convergence towards a phenotype that is more transmissible. And if the virus continues to circulate in an uncontrolled fashion, that could potentially become even worse. We're also seeing the emergence of strains which have changes in the spike protein that make it increasingly resistant to our countermeasures, both to our monoclonal antibody therapeutics and to our vaccines. And we now have real-world data from Novavax and Johnson & Johnson showing reduced effectiveness against the variant that first emerged in South Africa, this so-called B.1.351 variant. What is CEPI? So sharing the effective countermeasures that we have as quickly as possible to protect the most vulnerable is in everyone's interest for all of the reasons that I've just cited. What is CEPI doing in this regard? We have supported research and development. We've invested over $1 billion to support the development of vaccines, including we have worked closely with Curabac, who has a very promising vaccine, to make sure that the world has the tools that it needs. We have, using our R&D investments, which reflect the investment of public sector funds, we have secured access agreements. So the partners that we have partnered with, we have asked them to provide doses into the COVAX facility, which is committed to equitable access and to ensuring that this vaccine, particularly while it's a scarce resource, is shared globally and targeted to the most vulnerable. And finally, with our partners Gavi, the Vaccine Alliance, and with the World Health Organization, we have established COVAX, which is a multilateral effort. It involves private sector partners, it involves sovereign countries, multilateral organizations like WHO and UNICEF, coming together, bringing their individual competencies and skills together in an effort to create an end-to-end mechanism for developing vaccines, procuring vaccines, and delivering and distributing those in a fashion that is in accord with the principles of equitable access.
Thank you, Richard. It sounds like a big task to distribute globally. Can you give us a short hint about how this can be done? Don't you have to prioritize in any case, right? Can you avoid this?
It's an extraordinary undertaking. I mean, it will be the biggest vaccination effort ever undertaken in history. And it entails the distribution of a variety of vaccines on different platforms with different attributes, different requirements in terms of cold chain or administration, in some cases different intervals for dosing. There's a manufacturing challenge, there is a regulatory challenge, and there's a distribution challenge. And at this point, probably thousands or tens of thousands of people are involved in the COVAX effort at the country level, at the regional level, and working in the sort of the core planning group that we're part of to bring all of these pieces together. It's an absolutely massive undertaking. But I think the fact that we've had 191 countries sign up to participate also speaks to the desire for a global solution. And we've gotten to where we are by solving the problems as they come at us, and that's how we're going to solve all of the challenges ahead of us.
Mm-hmm. Sounds very impressive. Thank you, Richard. I'm now moved to our fourth member of the panel, which is Professor Heidi Larsson. She's also in London and director of the Vaccine Confidence Project at the London School of Hygiene and Tropical Medicine. Where COVID-19 vaccines are available, public uptake is essential. Experts say up to 70% of the population need to be vaccinated. A recent survey by the World Economic Forum shows that vaccine confidence has risen in countries where vaccines have started to be administered, while it has dropped in many other countries. You headed UNICEF's global immunization communication, and in 2010 returned to academia in order to investigate how rumors and misinformation affect global health. So my question is, what are the most important factors in promoting COVID-19 vaccine acceptance and uptake? Is it just a question of providing correct information?
Thanks very much. Well, I'll start with the correct information. I would say that correct information, accurate information is foundational and important for any communication strategy around vaccines, but it's not enough. And it may not even be, it may even aggravate some people because that's not their issue. So I would say the most important thing for vaccine acceptance and uptake is trust. And it's not just about trust in the product. In the Vaccine Confidence Project, we talk about trust in the product, the vaccine, trust in the provider, whether it's the local health provider or the producer, and trust in the policy and politics. And that's often where it tends to break down the most, is at the trust in government, particularly for marginalized groups who are less trusting of the government. And trust is really defined by two things. It's based on trust in the abilities or competence in an individual or institution. And it's the other dimension of it is trust in the motives. And that is often where it breaks down when it comes to vaccine trust, is question of the motives. Why are they giving us all these vaccines when that's not my real need? That's not my felt need. So I think from a communications perspective, from an engagement perspective, that's why we frame our research group as a confidence project. And it means that the communication needs to be dialogue. It needs to start with listening, because while the information is crucial, it needs to be relevant to the issues that people have. So you're not going to know what's going to be relevant if you don't know what the issues are. And sometimes they come out as being about rumors. And I just published a whole book with Oxford University Press called Stuck. And it's about this disconnect between the scientific community and the public, and where that misses the point in the middle. And we just need to make the relevance issue is important. For instance, yesterday there was quite a there was some coverage of the issue of minority Asian and Black communities having much less uptake in the current vaccine rollout than the white population. And one of the community members said, the problem is this communication, the things they talk about is when they close the pubs and whether a Scotch egg is qualified as dinner, meaning you could go or not. And that has zero relevance in some of these communities. So making sure that you use local, not just language changes, but things relevant to the concerns, because they also don't feel like it's meant for them. Work at home for a lot of people is not an option. How can we bring in more language that suggests that the government actually cares about people, and somehow embedding our vaccine information, particularly in the context of COVID, where people have so many other anxieties and stresses. We see a lot of correlation. We just published or are about to publish a large paper with 32 countries on willingness to accept COVID vaccine, and across all the socioeconomic and demographic factors. In addition to that, we asked about their people's perceptions of how the government was handling the overall COVID response. That was overwhelmingly the strongest correlate of positive willingness to take a vaccine, much more than any other socioeconomic or demographic factor. So if you want to, and we do in our surveys, we're asking about experience with lockdown, with masking, with other measures. So if you want to really get the whole picture and understand that relationship, it's important to ask a few other questions around that. I often say we don't have a misinformation problem as much as we have a relationship problem. because people, when they have trust, they'll put up with a little risk. If they don't have trust, they're always looking for there's something wrong. So if you build that trust, it's already building a bit of trust there, which is crucial in these times. The other challenge we have is one of the reasons that people sometimes refuse vaccines or question them is not because they have a particular issue with a vaccine, but they have another alternative belief, either pro-nature, homeopathic, anthroposophic, or religious. We have a number of issues sometimes with the Muslim community around whether there's porcine in the vaccines, like with the oral polio vaccine, or even here recently, actually a couple of years ago, in the Muslim community about the nasal flu vaccine because of the gelatin that adheres the vaccine in the nose. We thought it would be, on the one hand, we think that making it a needle-free vaccine will make it more acceptable, but the anxieties of some of the community members was that it was too close to the brain and might affect things. But they also had issues with the porcine in the nose, but actually the bigger anxiety was too close to the brain. So I think without asking people about these, it's difficult. And just the last point, aside from just stressing the importance of these trust relations and talking to people about how they're feeling in general, so they feel like you actually care how they're feeling, and then we can talk about the vaccine, is trying to fit the vaccine into their frame, into their value, into their beliefs in a way that doesn't focus, for instance, as much on the porcine in the case of the imams, and certainly with polio vaccine. We talked about, in your religious framing and your values, it's about saving lives. And there's no alternative. And that's certainly the Organization of Islamic leaders have said that when there's no alternative, then, it's legitimate. That doesn't always work for some locals, but making sure it's relevant to people's lives, I think, is the most important thing. Thanks.
Thank you, Heidi. That's a fascinating research area. We have more time to discuss, but after this first round through the panel, I would like to ask for the results from the poll to be presented. And the question was, would you get vaccinated against COVID-19? And 82% of our viewers say yes, they would. 10% said maybe. It's only 3% who said no, and 6% are already vaccinating. So we have an overwhelming majority ready to be vaccinated, but also some maybes. And so there's reason to offer more information and background on vaccination here. I would like to continue with a follow-up question to Gagandeep. There are new strains of the virus that have been identified, and some are said to be more transmissible. How do these new variants affect the immunization programs, and will the vaccines that we have be still effective with the new variants?
I think one of the things that's important to understand about RNA viruses is that they change, and they will and are expected to change with time. Early in the pandemic, we had a variant called D614G, and that was a variant that replaced the virus that had existed before. And then from around early December, we started to see news of other new variants, which had been around for a few months, but were beginning to take over the surveillance landscape in the UK. And part of the reason that it was possible to identify those variants was because there was a really strong surveillance program, a strong sequencing program that was in the UK that showed us that things were changing there. It seemed quite quickly from epidemiological data that the virus was spreading faster than the previous virus had. It was resulting in an increase in transmissibility of the virus. And that has now been borne out by some of the other new variants that are being seen in other parts of the world as well. So we have variants that have been identified in South America, in Africa, and in Europe. And the reason that we know that these variants are there is because sequencing is happening for those variants. It's very possible that there are variants in other parts of the world. Now, if we look at variants, there are really a number of reasons why variants are important. The first is diagnostics. Can you actually identify the variant is a variant, or will you lose the ability to test? And fortunately, that has not happened yet. We are still able to detect all viruses with the diagnostic tests that we have. The second is, does this virus spread more easily? And it seems like some of the mutations that we are seeing are resulting in a better ability of the virus to bind to its receptor. And that's resulting in increased transmission. The third is, does the virus, when it mutates like this, does it cause more severe disease? And we don't have clarity on that. There are some signals that that might be happening, but not particularly clear yet. The next is, will the virus actually be able to escape the treatments that we have, whether those are monoclonal antibodies or drugs? And there is laboratory data that seems to indicate that under pressure, the virus can mutate to escape certain monoclonal antibodies that have been developed. And the last and the most important one for this session is, can the virus escape immune responses that are generated by vaccines? And as Richard had mentioned, there are now real world data that tell us that in South Africa, we are seeing lower protection from the vaccines that have been recently evaluated there. And those are the Novavax vaccine and the single dose Janssen vaccine. What does this mean for the future? It means that we will have to think. We don't need to change vaccines tomorrow, but we will have to think in the future about changing vaccines, maybe do what Moderna is planning to do, which is give 1 dose of the older vaccine and give 1 dose of the new vaccine as a booster, and see if that gives better protection against the strains. I think this is important, but it's also important to emphasize that we have tools to deal with this problem.
Thank you, Gagandeep. I already receive questions from the audience, and there is a question to Florian von der Mülbe, which connects to the issue that we just discussed. So the question is, when new mutants emerge, can you add these to the already existing sequence to make it a polyvalent vaccine? Is this possible, or do you have to produce a separate vaccine for each new mutant? Gagandeep already discussed a part of this. What's your perspective on this?
I can mostly comment from the manufacturing part and not from the clinical part, as there are other experts. So from the manufacturing part, the change in manufacturing is very easily done. And we also, as Curabac, we have, as mutations are a natural survival strategy, as we heard, of virus, we have established a process to continuously capture and process information about any mutation that occur, and look whether this is covered by our vaccine, yes or no. And we can, from manufacturing side, easily introduce this to our existing production process. We could do two ways. On one hand, we produce it separately on different lines. What would be, I think, the way forward, as you have to have a very defined to produce a very defined product that you have to characterize, and this could be then given as proposed already as a second booster or whatever, or mixed directly together. So from the RNA level, mixing is not a problem, I have to say. Whether this is regulatory wise or clinically wise the way to go, this has to be decided and seen by data. So what would help us all, I think, would be a very lean way forward from regulatory side, how to introduce these new mutants, whether there always have to be additional typical clinical studies from phase 1 to phase 3, or whether it could be introduced more easily, as we know, for example, for a flu vaccine. So that's beyond the pure manufacturing, but very important questions to ask.
Thank you, Florian. Who would be in charge of changing these regulations? Would that be a question for the European agency, or is this a national issue?
So I think EMA, of course, is and FDA are there to approach, but I also do think that WHO could help on this, and I know also with the help of CEPI, they are very active in this worldwide harmonization, more or less, how to react. And I think CEPI is there a very important player who knows all the contacts and who to address for these kind of questions.
Mm-hmm. Thank you. Richard, do you want to comment on this, what CEPI has been doing here?
Sure. I mean, I think it is a complicated international regulatory environment. WHO, through its pre-qualification process for many countries, will offer the stamp of approval, essentially building on the evaluations of the stringent regulatory authorities that WHO recognizes. And those are agencies like FDA, EMA, MHRA in the UK, and a few others worldwide. There are also, just to mention, there is the ICMRA, I hope I get the acronym right. I think it's the International Council of Medical Regulatory Authorities, which I think is playing an increasingly important role in bringing global regulators together to look at these issues and to consider the issues. I think one of the challenges that's worth mentioning, a lot of people have looked at the flu paradigm, where there is a annual updating of the vaccines. That process normally takes 8 or 9 months between the development of the new vaccine candidates, the immunogenicity testing of those candidates, and the regulatory approvals. 8 or 9 months is simply too long. We have articulated, and many of our private sector partners have embraced the idea that we should, from the decision to develop a new vaccine candidate, we should be able to make that candidate available within 100 days. And to accomplish that, we would have to have very tight coupling between the efforts of the companies developing the new vaccines and their manufacturing components with the clinical trials to test those candidates, and with regulatory guidance, which is clear from the very beginning as to what will be required, and allow for a rolling regulatory review if we're going to achieve that 100-day goal. But that's absolutely essential if we're going to respond and stay ahead of this virus, which is moving a lot faster than flu.
Mm-hmm. Thank you, Richard. So from confidence for immunatory hesitancy, does it make a difference for individuals whether there are mutants? Does it affect the acceptance of immunization? Do you have any evidence on this, Heidi?
Well, it certainly is creating some hesitation or an anxiety. I think we don't have a specific, I mean, we're at the kindergarten stage when it comes to rolling out vaccines. And I think we're really going to see where the tough parts are by the end of the month into next month, when we're going into the broader population and larger numbers. But right now, just in terms of our surveys and listening, indeed, it is an anxiety, adding to the list of people wanting to say, I'm going to wait for somebody else to take it. But it hasn't turned in, I mean, people aren't saying, I don't want it. They just want to say, if I'm going to take that risk, I want to make sure it's going to work.
Mm-hmm. Yeah, thank you. I have a long list of questions to ask you. I would like to continue with Gagandeep again. There was a question about whether vaccines also prevent the transmission of COVID-19. What's the state of knowledge on this?
If we look at what we want to assess for vaccines, we want to see first and foremost, do they protect against severe disease? And within that, do they protect against any disease at all, right? So that's the first thing, and that's the primary outcome for most vaccine trials. After that, you want to look at, do you get a decrease in infection, and do you get a decrease in transmission? If you achieve a state that is called sterilizing immunity, which is where if you give the vaccine, that person cannot be infected, then you will prevent transmission as well. But it's possible that you may be able to prevent disease, but not prevent infection, but still restrict virus replication to a much lower level, so that overall there's less virus around and less virus to spread. So within some of the vaccine trials that are being done, I think we will get some signals about the level of virus that is being shed by people who have been vaccinated and are still infected. And that will give us more clues that will help us understand what is going to happen in populations, not just at the individual level.
But so far that's not known, right? That's not something that's checked in the first round. That's something that's a secondary issue to be addressed.
It's a secondary issue, and it's not in the published data as far as I'm aware. And Richard may have more information on that.
Richard, do you?
Just a couple of things to add. I think we do need more data on it. The AstraZeneca trial, they did look, they did have the participants in the AstraZeneca trial come in and be tested for infection, I believe on a weekly basis. That's the only trial that has followed patients that intensively. Most of the other trials have looked at symptomatic disease, clinical disease. The AstraZeneca study did show asymptomatic infection in some participants. The other piece of evidence I think that's relevant, and there's emerging epidemiologic data looking at rates of secondary attack rates with documented infection but asymptomatic infection versus secondary attack rates with, and this is principally looking in household where people are documented either to have asymptomatic infection or clinical infection. The secondary attack rates are higher with clinical infection. So I think there is some logical grounds, as Sherry Gagandeep was saying, to think that if you reduce the severity of disease, you may actually also see some concomitant reductions in disease transmission, but that's a hypothesis at present. I think we need to really demonstrate that conclusively with data.
Thank you, Richard. I now have a question more on the production side for Florian, and the person wants to know, when new mutants emerge, can you add these to the, oh no, sorry, that's the wrong question. The question is, where do you get your mutants for research, and how do you decide which ones to use as a promising template? So what's the origin of your data that you work with, and how do you decide on which issues to focus when you work with the mutants?
Mm-hmm. So the data is a great network, I have to say, about databases coming from different countries where our bioinformatics look always and screen, and out of these processing, they give recommendations that then are also checked if necessary. So we are again, we are not dealing with the whole virus or something like that. So we are only looking at certain structures or certain antigens. So that's not that we have all viruses here in house or something like that. Absolutely no. The RNA technology is only building on this small pieces of this genetic code. So that's how we approach it. And therefore our sequence analysis is most important. And if necessary, we try really to check in other studies whether it's still covered or not. But in the end, also clinical studies have to be conducted, and there the entire community has to work together, I think.
So you try to find a consensus on what's the specifics when you work with specifying the virus type that your vaccine work against.
Exactly. So we have, of course, the spike protein, and we know a lot about the language of RNA, so to say, and we try to simulate as best as we can whether we have hints for escape and so on, and to then also design, if necessary, new product candidates to target these, if necessary. But as it was already said, fortunately, there is still a coverage by the existing vaccines.
Okay, thank you. I have a question that goes back to Richard. Discusses the second element of your task, the equitable provision. So the question is, with the global competition on COVID vaccines, is it really feasible to make vaccines available in poor and rich countries at the same time?
Well, I was informed this morning that we just passed a significant threshold, I believe today, where the number of doses of vaccine that have been distributed actually now exceeds the number of confirmed cases of COVID that have been recorded. So we have 104 million doses of vaccine that have now been distributed, and 103.5 million, I think, cases of confirmed COVID. The very vast majority of those 104 million doses have gone to high-income countries, as I think everyone is aware. So we have not achieved the goal of distributing vaccines simultaneously. But COVAX is planning to distribute the first doses of vaccine to many, many countries starting this month, and we are on track to deliver, I believe, around 465 million doses by the middle of the year, and over 2 billion doses by the end of the year, which is towards our goal of making sure that every country can vaccinate to at least 20% of their population to protect their healthcare workers and their most vulnerable populations. I think it's relevant to think about historical precedent in this regard in terms of sharing life-saving medical interventions. We didn't do it simultaneously, but if we start delivering this month, that will be within 2 months of the initiation of vaccine deliveries to high-income countries. With HIV therapy, highly active antiretroviral therapy, those became available in high-income countries in the late 1990s. Death rates from HIV continued to climb in Africa for more than a decade after those therapies became widely available in high-income countries. In 2009, during the last pandemic, the H1N1 or swine flu pandemic, vaccine became available in developed countries in September of 2009. The first doses under a vaccine donation program were distributed to 4 countries, just 4 countries in January. Those were the first doses to reach the developing world. And a year after the peak of the fall wave of the 2009 pandemic, only 78 million doses had been distributed. So I think we are moving the bar forward significantly, even if we didn't achieve the goal of simultaneous distribution.
And do producers give up some of their profits, or is this just the money raised by donations? Or do prices go down over time anyway? So what are the key mechanisms there?
Well, in terms of how we're negotiating with the manufacturers right now, we have groups that are receiving vaccine through an advanced market commitment, and that has required donation of funds from high-income countries. We also have self-financing participants that are both high-income countries and upper middle-income countries. So we have actually 3 tiers of income categories, and we are securing vaccines at tiered pricing. So there's a high-income country price, an upper middle-income country price, and a price for the AMC participants. Over time, as manufacturing is optimized and as production capacity increases, we believe the price will continue to come down. But right now, we're using tiered pricing as our mechanism.
Okay, thank you. I also have some questions for Heidi from the audience. One question is about the role of general practitioners. Which function do they have in creating acceptance and trust in the vaccines, and how could the GPs be supported?
Well, one thing that would be helpful is if the GPs themselves have been vaccinated, because one question that people are going to ask is, have you done it? I mean, have you taken the vaccine? And we are seeing some hesitancy among healthcare professionals globally, but across Europe, we have already been working closely with the EU for the last 5-6 years, monitoring vaccine confidence across Europe, and last year had already added some investigation of healthcare professionals. I know it's also a global issue in India. I believe it was in Gujarat state. Correct me if I'm wrong, Sherry, but it was nearly 80% of healthcare professionals in one state that did not want the H1N1 vaccine. And that's a much more familiar, unknown virus, a much more familiar preparation. But, and in this case, we're facing more. So from the GP perspective, if they have an opportunity to get one, or at least express an intention to, that will help. But I think interactions with GPs are again an opportunity to listen to the concerns of people. And I think by helping alleviate their concerns will be important. But I do think getting information to GPs and having a forum for them to be asking questions, because they are not immunologists or vaccine developers, and it's all new for them too. So the more we can get them access to information as it becomes available, the better.
Yeah, that makes sense. I also have a question for you on what governments can do. So we talked about the GPs, but if there is distrust in governments, what can the government do to improve the trust in vaccines? Are there any established procedures that you believe work and have not been used by every country? Is there anything that we can learn from past experience for government interventions?
Well, I think from a government perspective, the more that they can diversify their messages and their outreach to be adapted to different settings, the better. If it's only in the mainstream core language and not adapted to local languages, not, as I mentioned earlier, recognizing the different issues, cultural context, and when it comes to COVID, if you're talking about changing behaviors, recognize what are those behaviors. So I would say have a diversified, locally adapted outreach program involving and engaging local community leaders, local clinics.
Yes, thank you. Then I have a question jointly for Gagandeep and Florian, maybe. There's a discussion about delaying the second jab. Can you say something about the effect this has on the efficacy of the vaccine? Is it the same for all different vaccines? What should we consider when we talk about delay, Gagandeep?
When we look at the data from the AstraZeneca trials, which is what this variation is based on, it seemed like when there was a longer interval between doses, you got better efficacy of the vaccine. There's also more recent data that has been published that shows that even a single dose of the AstraZeneca vaccine does give reasonable protection. So the arguments are twofold. One is an immunological argument. Do you actually make a better immune response if you separate the doses of vaccine? And going back to childhood immunization schedules, we know that in the Western world, there is a 2-month gap between vaccines. So vaccines are given at 2, 4, and 6 months in some countries. Whereas if we look in developing countries, we give the vaccines earlier at 6 weeks of age, and then we have shorter gaps of 6, 10, and 14 weeks, only a 4-week gap. Why is there a difference? Well, actually, you get a better immune response with the more stretched out schedule. But in developing countries, you have to balance the immune response with how early disease occurs. So we have a basis to say that immunization schedules don't necessarily need to be exactly as they are laid out when you do a trial. For the AstraZeneca vaccine specifically, an argument has been made that because this vaccine results in not just the production of the spike protein, but also the display of the spike protein on the surface of the cells that are infected, the longer interval might result in a better T-cell response to the vaccine. So the argument is if you go out to 12 weeks, and there's some work that has been done with adenoviruses for HIV vaccine development, that seems to indicate that a longer interval might be beneficial. That has to be balanced against what virologists think about, which is if you don't make a really robust immune response with a vaccine and the person gets infected, then are you giving the virus more time to evolve and have vaccine escape? So both of these need to be considered, and that's the biology angle. Then there's also the public health angle, which is how do you get most people vaccinated? And you can do that if you give a single dose first and then follow up with a longer interval as vaccine becomes available. So it's complicated.
Yes, thank you, Gagandeep. Many different dimensions. Florian, would you like to add on that?
I think Gagandeep really described it very comprehensively, and it's always difficult to assess without data and try to find this balance. So second dose seems to be necessary for a boost or whatever, but it coming a lot of different angles together to decide on.
Mm-hmm. Thank you. I have a lot of additional questions, but our time is running out. So I would like to give each one of you a minute for a concluding statement where you may sum up your main message, or what would be your advice for policymakers, or what's your take-home message for our audience. Maybe I start with Gagandeep. What are the important points in your view?
I think the most important point is science brought us solutions and we got here by working together. So the lesson for the future is just that we need to continue to work together, do science without borders, and we need to think about the world as a whole, not just our corner of it.
Mm-hmm. Thank you. Florian, you are the second. What would you tell the audience or the policymakers?
Okay, I think that it's really, the pandemic is a terrible thing, no doubt about that. But as Gagandeep said, it also opens some doors, I have to say, how to work together, and we should all try to use it and find their together solutions. And I'm convinced that we have tools in our hands, as for example, the RNA technology that could easily be adapted to certain needs. On the other hand, how to include companies here, of course, it's always important to plan for the future. And I think what was underestimated in the past, and therefore also great thanks to the CEPI initiative, how to prepare for pandemics, and perhaps also do here an investment in advance, and also give a chance to new technologies, as RNA was it in the past, but take here also risk and money to take this very serious, that also future pandemics could come. But we have tools, and we can stand together and find solutions.
Thank you. Richard, your takeaway message.
I want to build on what the previous two speakers have said. I think science is our most powerful instrument in the fight against the virus. It helps us to see what is coming. It gives us the critical information that we need to apply the tools that we have. I think there has been a period in the response to the pandemic that has been characterized in many countries by wishful thinking. And I think we shouldn't confound optimism about our ability to respond and to defeat the virus with wishful thinking. And I hope the period of wishful thinking is over. It's science that should make us optimistic, and it is science that will lead us out of the pandemic. One final comment related to equity. I think if there was ever a period when the idea that we could get out of the pandemic simply through vaccinating our own populations in our own countries and leaving the rest of the world to, sort it out themselves, the emergence of the variants make it very clear that the defense of just first-line vaccination may not be sufficient. We can only get out of this pandemic by working together and by sharing the tools we have equitably.
Mm-hmm. Thank you. Heidi, what is your bottom line?
Well, I think science is absolutely crucial, and also social science in all of this, which I think is the science that can bridge the public and the technical experts, clinical and discovery end. But I also think that COVID has been an absolutely huge opportunity for people to have they've never been so exposed to science around vaccines. Its breadth, how many people knew before COVID how long it normally takes to develop a vaccine. So this is a huge opportunity to build public awareness and trust in vaccine science, and really hopefully change the landscape moving forward in that understanding.
Thank you, Heidi. So yes, I think we have learned a lot in our conversation and had a chance to look at vaccinations from various perspectives, from the perspective of science, from the perspective of production, from the perspective of global administration and acceptance. The pandemic poses a challenge to humanity, but we are fighting back and we are using science to do this. I hear a lot of optimism in your communication and the idea to work together, I think, is also an important message of this panel. I would like to close the panel by thanking the panel members for participating and for providing their expertise, and by thanking the organizing team for setting this up so professionally. And also I thank the participants for the many questions in the chat, and I hope we jointly fight the pandemic successfully and get together and improve science meanwhile. Okay, have a nice day. This concludes our panel discussion. Thank you. Bye-bye.